Hey, you guys, here we go. Just getting you guys started in some notes for this will be probably the second to last set of notes that you'll need to take with evolution and genetics. And so today, this stuff will be for Wednesday. So I need you guys, please be sure that you are taking notes on this stuff, as this will be found on the test that's coming up next week, at the end of next week, and also the final for the week after that. Okay, and so please make sure that you guys are taking notes on this stuff. But today, basically, what we're doing, last time, well, I guess 2 days ago, I got you guys started into natural selection, and hopefully that was a review of what natural selection is and how it can cause change in an environment. And specifically, I hope you guys are in the lens of genetics and see how this can cause a change in the genes of the population of that organism that we're looking at. How we do this, and you might have some experience with Hardy-Weinberg. It just depends on the teacher, I think. But anyways, how we kind of look at this and how we can kind of analyze the data is using this principle, and it's called Hardy-Weinberg equilibrium. And so there is going to be some math that you're going to be doing here. It's not too bad, though, and I'm going to walk you guys through that and show basically the concepts behind Hardy-Weinberg and how it shows and models natural selection. Or not just natural selection, but how it shows evolution occurring in a population. So here we go. Let me pull this up there. Alrighty. So to get started, sorry here, just getting everything set up on my end. Okay. So the first thing, the Hardy-Weinberg equilibrium can be used to test whether a population is evolving or not. And so with this principle, we can use this concept to say, is this population that we are analyzing, that we're observing, is it evolving or is it not? And if it is evolving, then we would say it's not following Hardy-Weinberg equilibrium. If it is not evolving, then it would follow Hardy-Weinberg equilibrium. And so you guys can start to see the statistics that we can do here, looking at at a population and determining, using the Hardy-Weinberg equilibrium, whether or not this population is evolving or not. And to do this, let's just do a quick review. And so this comes from last lecture that I gave, that I recorded, and it is found on Schoology. So if you need to go refresh, you can. But basically, a population is a group of organisms that are capable of breeding and producing fertile and viable offspring. And in that lecture, I kind of talk about, well, if you have a horse and a donkey, they can mate, and they produce a mule. The mule, though, is not fertile. It cannot have offspring of its own. And so this is an indicator that those two are not of the same species and therefore are not of the same population. Okay. And so, however, if you have a horse and a horse, they can mate and their offspring do produce fertile, or their offspring are fertile. They can produce viable offspring. And so we would say that these two horses are of the same population. And then with a gene pool, when we talk about gene pool, we're talking about all the genes, all the alleles for each gene that are found in a population. And so a gene pool is massive. And the bigger the population size, the bigger the gene pool, because it's found for all genes. And then we remember, for each gene, we have different alleles. Okay. But then we would have to multiply that by each organism that makes up that population. And so doing that, we can have a massive gene pool here. And so let's kind of dive in and see, how does Hardy-Weinberg affect a population and the gene pool of that population? All right. So what does it look like when a population is not evolving? It could be a question here, because remember, with Hardy-Weinberg, we're looking at is it evolving or is it not? So if it's following Hardy-Weinberg, we would say it's not evolving. And so what would that look like here? And if a population is not evolving, then what we would actually see is the frequency of alleles, the frequency of genotypes be the same over one generation to the next. And so we could say, for example, I'm going to use our flowers that we're growing that are not germinating as well as I wish they would. Back there, I saw there's a couple cups that are, but anyways, a couple of them aren't doing so hot. But anyways, when we're looking at that, remember that we have, for example, let's just use the letter A here. It's going to work. There we go. So we have A and lowercase a, and let's just look at one gene for this. And so let's say that, for example, this gene that we're looking at is stem color. I need my wife's Surface Pro would make this kind of sweet. Anyways, but we have stem color, and we know that we have one allele. The dominant allele is for purple stems. And lowercase allele would code for green. So we have two alleles. We have dominant and recessive. But then we remember we have several genotypes that we could have. So right here, these ones would be purple in their phenotype, and this one would be green in its phenotype. And so we would call this homozygous recessive, homozygous dominant, and heterozygous. And so when we're looking at this, when we're looking at that population, let's say that Gen 1 had well, based on the data that we got, we saw that 100% were purple. Now, do we know which one? According to our data, we don't. We will after Gen 2 comes, or the F2 generation. But anyways, with the F2 generation, if you look, we don't have a lot of data yet, but let's say that it follows a ratio of a 1 to 2 to 1, where we have 1 being homozygous dominant, 2 being heterozygous, and 1 being homozygous recessive. And so we can see that this frequency is changing. According to this one, let's say that this one was a 0 homozygous dominant. All of them were homozygous recessive, and none of them were homozygous recessive. So all of them were heterozygous. And so we can see that this frequency, it changed from generation 1, where we had 100% heterozygous, while here we have a 50% heterozygous. And so we see a change in frequency and in that genotype. We could then look at the individual alleles and say, well, how many dominant alleles are there in this population? How many recessive alleles are there? And so we'd have to count how many individuals, and we'll dive into this a little bit. But this goes into that concept of, did it stay the same or did it not? And so based on this information, we could use Hardy-Weinberg to determine and say, guess what? This population is not it's not staying the same in each generation. So therefore, something's going on. Okay? All right, so in order to solve that problem, we need to know how to calculate these frequencies of alleles. And so that's kind of what I was going into there, and I kind of went on a little tangent, and I'll try not to do that so you guys don't have to sit here and listen to my lectures much. But anyways, for a diploid organism, the total number of alleles at a gene is the total number of individuals times by 2. Okay, because remember, each organism will have 2 alleles for each gene. So here's like kind of an example. You could be homozygous dominant, you could be heterozygous or homozygous recessive. No matter what, though, you always will have two alleles for that gene. And so, for example, if you are homozygous dominant, what do you have? We have two alleles that are dominant. If we are heterozygous, though, what do we have? We have two alleles. One is dominant and one is recessive. If you're homozygous recessive, though, you have no dominant alleles and two recessive alleles. And so we can start to add this all up and calculate frequencies. And so to do this, you can kind of use this equation right here. And so, for example, let's say we're looking at the dominant allele. We would say that the frequency for the dominant allele is the total number of dominant alleles in the population divided by the total alleles in the population, both dominant and recessive. And when you do this, and you calculate the frequency of this, the frequency of both the dominant and recessive allele should equals 1. So right here is this equation, and make sure you guys write this down. And so what we kind of do when we're looking at Hardy-Weinberg is they use P and Q. P is going to represent the dominant allele. Here, let me actually erase that so it's going to represent the dominant allele, and Q will represent the recessive allele frequencies. And so P is the dominant allele frequency, Q is the recessive allele frequency. And when you calculate these frequencies, add them together, you will get 1. So for example, I've got actually an example coming up, so we'll just wait until that. But make sure you understand this. So 1 is also it's also talking about it being 100% total. All right, so let's look at an example here. So first of all, let's consider a population of wildflowers that show actually an incomplete dominance. Okay, so they're not complete dominance, and so when we have incomplete dominance, we would have homozygous dominant would have a phenotype. Heterozygous also has a distinct phenotype, and homozygous recessive also has its own phenotype. And let's say that in this population we have 320 red flowers, 160 pink flowers, and 20 white flowers. The first thing I want you guys to do, and I'll let you pause, and I want you guys to try and calculate this. But I want you to calculate the number of copies of each allele. So I'll ask the sub to pause the video. I want you guys to attempt these, and then when you're ready, push play, and I'll work these out with you. Okay, so I'm going to work these through. I'm assuming that you've attempted these. So first of all, how do we calculate the number of each allele? And so when we're looking at this, we would basically have to count how many alleles we have. And so first of all, what we're going to do is we're going to look and see, okay, well, we have CR, which is our dominant allele, and we have CW. So let's figure out how many total alleles we have in this population. So CR, we have one right here, one right here, and one right here, and we have 320. So 320, we're going to actually times this by 2 because each one of those organisms has 2 alleles. So we would actually have 640. But we're not done yet. We also have one found right here. So we're going to actually add 160 to that, which that equals, let's see, so that'd add up to 100, so 6, 7, 800. I think I did the math right in my head. Yeah, 7, 8, yep, okay. So we have 800 alleles that are of this allele. Now let's figure out how many we have of CW. So right here we have 20, so we're going to have 40, because we're going to multiply that by 2, because each one has 2 organisms, or 2 alleles of that kind, and then we're going to add 160 to that. Thank goodness for my math here and the simplicity of it, but that adds up to 200. So we've now calculated the total number of alleles. The next thing I want you guys to do is calculate the frequency then. Remember, we're going to use, when we're done, P plus Q should equals to 1. Okay, so I'll let you guys pause the video, try and calculate using that equation that I showed before, not this one, but how to calculate the frequency of an allele. Try and solve these. I'm going to keep going. I think you guys have paused this, you've solved that. And so doing that here, what you can do, and I'm going to pull out my calculator for this one. Here, I'll pull up the calculator so you guys can see me do it. Let's see. All right, so here's the calculator. So what we need to do is we need to calculate the frequency for CR and CW. Ooh, there we go. And so how we'll do that, I'll just draw it out right here and then we'll pull out the calculator again. CR, we have 800, so we're going to take 800, but we're going to divide it by the total sum, which is 1,000. 800 plus 200. For this one, it's going to be the opposite of that. So 200 divided by 1,000. And if we do this right, both of these values, when added together, should equals 1. And what we're going to notice, this is going to be our P value, and this is our Q value. So here we go. Let's pull up the calculator. Okay, so we have 800. We're going to divide that by 1,000. 0.8. I don't need the calculator for this. And what's this one going to be? 0.2. Okay, and if we add this together, 0.8 plus 0.2, does it equals 1? Yes, it does. So we're good. Okay, and so you can see how we can use these values to calculate these frequencies. These frequencies are important because it's going to be used later on. So let's dive into this. So the Hardy-Weinberg principle basically states it's a description of whether or not that population is evolving or not. Now, if it meets, like I was saying, if it meets Hardy-Weinberg equilibrium, we would say that it is not evolving. So if a population does not meet the criteria, then we would say it is evolving. And the Hardy-Weinberg principle states that frequencies of alleles and genotypes in a population remain constant from generation to generation. Okay. However, like I said, if it's not staying constant, then we would say it's evolving. Okay. So make sure you understand that those key parts there. So let's look at this and let's try and solve this. So we're going to use this equation, these values that we looked at. So consider the example from the population. So we just did this, and these are our values that we got, right? Now, if P and Q represent the frequency, blah, 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 then we can kind of put this into the Hardy-Weinberg equation. So I want you guys to write this down. You'll need to know this. Here is the actual equation for Hardy-Weinberg. And you can see we're using P's and Q's. P is your dominant allele. Q is your recessive allele. And when we're diving in this and you're looking at this equation, it's kind of fascinating. And that's why I have this picture right here. So look over here. So you have P squared plus 2PQ plus Q squared equals 1. What they've actually been able to do with this using math is the P squared is actually showing your homozygous dominant genotype, while 2PQ is showing your heterozygous, and Q squared shows your homozygous recessive. And this is kind of neat because then we can start to do different things, and that kind of makes sense. It makes sense here because when you're homozygous dominant, remember you have 2 dominant alleles, so it's P squared. You have 2 of those alleles. If you're heterozygous, you have 1 of each, so you'd be 2PQ. And then Q squared would be your homozygous recessive because you have 2 recessive alleles, 2 of those. Now when you add those values together, it equals 1. So with Hardy-Weinberg equilibrium, not only can we calculate the frequency of alleles, but we can also calculate the frequency of the genotypes. And with the genotypes, we can actually calculate the phenotypic phenotypic frequencies as well. Okay, because depending on this one, for example, each one of these is its own phenotype. If it was complete dominance, we could say, well, these two are one phenotype and this is the other phenotype. And so the sum of these would be one phenotype and this would be the other phenotype. Okay. And so this kind of shows you models of how you can do this. If P was 0.3 and Q is 0.7, we can calculate those frequencies. So what I want you guys to do using this information, I'm going to let you guys pause the video, but I want you guys to calculate the frequencies for genotype and phenotype using the values that we calculated with the flowers. So I'm going to assume that you pause the video and you are ready to move on here. So first of all, let's calculate the frequency for P squared, which is your homozygous dominant. What we would do is we would take this value, so 0.8, and we're going to square that. And whatever that equals will be our frequency. We're going to do the same thing for our heterozygous, so 2PQ. It's going to be equals to 2 times by P, which is 0.08, times by Q, which is 0.02. And whatever that value is, we're going to figure that out. And then we have Q squared, which is 0.2 squared. All right, so what is 0.8 squared? Well, let's pull up the old calculator again. So 0.8, I'm just going to do it the old way and just multiply that by itself. So we actually see 0.64. Then 2PQ, it's going to be 2 times by 0.8 times by 0.2, 0.32. I should do this right and put a 0. Okay. And last but not least is our 0.2 times by 0.2, 0.04. Now, if we did this right, the sum of these should equals 1. So let's see if that's the case. Okay, so our first one is 0.64 plus 0.32 plus 0.04. Boom, we did that right. Okay, so you can kind of see how that kind of works there. Now we can calculate the phenotypic frequency. Well, we kind of already did because each one of these is their own phenotype. Because it's incomplete dominance, and so there would be the same phenotype. You can then multiply these by 100 to get the percentage, so you'd have 64%, 32%, and then 4%. And so using these percentages, you could then say, well, if we have a population of these flowers, let's say we have 1,000 individuals in that population, which we then calculated, we can then determine how many would have each genotype. Or we could go into and say how many alleles, dominant and recessive, are there in that population of that. So hopefully that kind of makes sense how we did that, because you guys will be doing some problems with that. And so if you need to, I will also put this lecture on Schoology, and you can watch me go through that over and over again to your little heart's content. So anyways, here we go. So we can also use Punnett squares to model this. And so how they do this is that kind of the idea here is that both the mom and the dad would have the opportunity to pass on, because it is random, have the opportunity to pass on both a dominant and recessive from that population, and it would follow those frequencies that we would calculate. And then using that, we can multiply. So 0.3 times 0.3 is 0.09, and 0.7 times by 0.3, 0.21. And then what we can do is add these together to get the total value for that. And that is where that comes from when we're looking at P squared. There's just the one. 2PQ, well, there's two of those. Okay, so kind of interesting there. And then the one again for that. All right. Just one thing too is right here. So this is like an example of our gene pool. Okay. And then looking at that out of the total, we could then calculate those frequencies. So, all right. So here's another example that I want you guys to work through, and there will be several questions on the test, on the final, and in your homework or on your little work assignment today, where this is what you guys will be doing. And so you have sampled a population which you know that the percentage of the homozygous recessive genotype is. So I give you some values here. So now what I want you guys to do is using that information, trying to answer these problems. And so I'll let the sub pause, and you guys can work through these, and then I will work through them as well afterwards. Okay, so here we go. Let's dive into this one. This one's kind of fun. Using our equations that we now know, we can do this. And so there is a missing piece for this, though. We don't know. So first of all, we need to know our p-value and our q-value. With these, we can solve anything that will be asked. However, this is all we've got. Okay, and so we know that our homozygous recessive is 36%. So we know. So how could we solve for this? First of all, we need to figure out what Q is. How could we do that? Well, remember, we have P squared plus 2PQ plus Q squared equals 1. And we know what Q squared is because this is what that is. And so what we could do is we know that Q squared equals 0.36. Okay? Take the square root of that to get rid of the square. We can then solve for Q. So Q is the square root of 36, which is, oh gosh, me and math today. .6. Okay. So we would have 0.6 here. 0.6 times by 0.6 is 6 times. Yeah, that makes sense, Mr. Crip. There we go. Okay, so we have 0.6 here. So we know what Q value is. With this, can we then solve what P is? Absolutely, because we know that P plus Q equals 1. And so what we're going to do really fast, sorry, you guys have to pause. My kid just woke up. All right, here we go. So to you guys, this is just a split second in time, but my kid just woke up and I had to go put him back to sleep. So anyways, here we go. Let's keep going. So I think what we were at, we have P plus Q equals 1. We now know what Q is, and so we can solve for 1, or solve for P, sorry. So we know, so we could rearrange this equation. We know that P is equals to Q, or 1 minus Q, okay? And so 1 minus 0.6 is going to be 0.4, okay? And so we know that P is equals to 0.4. With that, we can now calculate everything else. And if you calculate that properly, this is what you should have gotten. Let me just pull everything back up here. Is this, okay? And so make sure you understand how to use those concepts there. So the next thing I want to dive into is this idea of with Hardy-Weinberg equilibrium, for it to be met, there are 5 things that need to be, 5 criteria that have to be met in order for Hardy-Weinberg to work. And the idea here is that if a population is not evolving, there should be no mutations. Okay? There should be random mating. There should be no gene flow. There should be a very large population size, not small, and no natural selection should be occurring. Okay, so for Hardy-Weinberg equilibrium to be present, that's what we would observe. And so we're going to dive into these a little bit more, but make sure you guys write those down. Make sure you know the 5 criteria. Those are big. So here we go. So the first one is mutation. So we know that mutations can cause genetic variation. We've learned about that. We've spent a lot of time on the mutations. And so, for example, if we have a frequency for the allele A is 1 and we have a mutation, now what we've done is we've introduced one allele of a different or one different allele into this population. And now it has the opportunity to change that population. And so, because of mutation, we no longer see the frequencies the same as what it was at the original. And so, therefore, it's no longer following Hardy-Weinberg equilibrium because evolution is occurring here. Okay, or it possibly could because of this mutation, and now opens up the opportunity for nature to work its magic. When we're looking at gene flow is a little different. What it is it's other alleles entering into the population, and how they can do this is it's whether it could be just movement of individuals entirely. So here's this is an example of a little beetle goes over this hill that was a barrier between these two populations, and now what we have is this one little dude here is now part of this population that was purely Q, while this one was purely P frequencies, right? And so now this one no longer is pure in it in being homozygous recessive. That's one way. The other way is you could have one of these little dudes come over here. Maybe they meet in the middle. They have babies. Okay. This one goes back. This one goes back. And now you've introduced alleles into those populations because of sexual selection or sexual reproduction. Okay. All right. The next one, when we're looking at the population size, this one's kind of interesting. I think it's kind of fun, but it's there's different types. But the first one is genetic drift. When we're looking at genetic drift, we're actually looking at population sizes changing and becoming very small. And this can happen due to chance events such as natural disasters or human effects that we have. For example, the buffalo, when the Native Americans were here, buffalo were all over the place. However, when the white man came and brought its smoke stick, its gun, we shot him down and put them almost to extinction. We changed the population size, and when we did that, the frequencies are more drastically changed. Okay, and so here's an example of, for example, if we were to take, for example, due to a natural disaster, these three organisms kick the can. The frequency now has changed pretty suddenly to not this yet, but we do have that. Oh, sorry. I'm on a wrong example. Let me erase. They don't kick the can. And so actually what we see here, let's say that these three organisms leave their little nest and they get separated from the other from this original population. Well, it was due to chance. This guy didn't go with them. Now this population, when it reproduces, it's only got recessive alleles to pass on to its offspring. And so that is all we would see. And so we see a what we call fixation event occur. And there are some labs that we will do when I get back looking at fixation. Fixation is no bueno. We don't want that. This decreases genetic variation. You guys love my handwriting. Anyways, it decreases genetic variation. Genetic drift does. It limits the population, makes them less fit to survive. And so changes in allele frequencies due to chance event. Drift can occur in any population, but has a stronger effect on smaller populations. So here's kind of another example of this. So here we have in generation one, these flowers, we see that we have a pretty standard allele frequencies. 5 plants leave offspring. And so we see it change to a 50-50. But only two, again, reproduce. Something's happening here, and these flowers are not able to reproduce with each other. And what do we see happening over 3 generations? We see fixation occur, in this case to the dominant phenotype or dominant allele. And so that's all. Now what we see is this entire population. These flowers are now dead. They can't pass on their alleles. They were less fit for some reason. All that was able to pass on is this allele. And so we see this fixation event now occur. So then if the environment changed and now these guys are not fit for the best fit, and let's say that a white flower would better blend into their environment or better survive, guess what? This entire population goes extinct because it does not have that opportunity, those alleles in its gene pool to expose or to use. So there's some different types of genetic drift that I want to talk about. One of them, it's called the bottleneck effect. I want you guys to make sure you know the basics between these. So just make sure you know bottleneck effect. What this is it's a sudden reduction in population size. So they use this bottle to model that here is some type of thing that all these aren't getting through. Only so many are, and so we have a small amount that survive. And so this is what I was getting into with natural disasters, such as a hurricane or a tornado or mudslide or something like that could potentially wipe out massive amounts. Say we start with 1,000 individuals in the population, could bring it down potentially maybe to 100 or even smaller to 50. And when we do that, we're now letting genetic drift take its effect, where it's due to chance alone of what alleles get passed on. And so you can see right here, it was random. We have 1, 2, 3, 4, 5, 6, 7. 7 of them were blue, and then only 2 yellow, when probably at first it was a 50-50 frequency. Do you see how fast that changed? 1, 2, 3, 4, 5, 6, 7, 8, 9. Anyways, we could probably maybe say it's like this. Okay, and so it changed drastically. Here's another one. Founder's effect. This one is kind of so they've actually seen this in human populations even, but basically it's when here we have this major population, maybe like for example the United States, a massive population of humans, and then let's say that a little family decides it wants to, or a couple families decide they want to ship off and go to a far, far away island. No one knows they left, maybe, and they're going to stay on that island and colonize it. And so, what they'll actually see is all they were able to bring were the alleles in that family. The gene pool now is significantly smaller, causing genetic variation to decrease. And then, when they start to reproduce with each other, not only are you having incest going on, and what that's causing. So, Lily, this is all for you guys. Lily, Gracie, I know you guys wanted to talk about this, but it's because of this idea of why incest is so bad. And so, the founder's effect, we've actually kind of done this with royalty. They only will make their spouses, they choose their spouse, and it only comes from a small population size that that they'll reproduce with. And so what we actually see happening is a small population, that those are the only alleles that it has to pass on to its descendants, to its offspring. And what we see is fixation occur, where it could occur more easily. And again, we're going to dive into this because I have several models and labs that we'll look at to show this example happening. But again, it's because of this that incest is bad. It decreases genetic variation. It allows for strange diseases to pop up that were maybe hidden because, for example, there's some diseases that you have to be homozygous recessive, right? And if the family was maybe heterozygous, all of them, you're bringing both alleles. But now the chance of reproducing to make this goes up significantly. And then as we see over time, you'll have some individuals have this, they could mate with somebody else like that, and now you're making the population change, that the allele frequencies are going to change very quickly. And again, I have labs that will model this, but anyways, little tangent. All right, so the last one, remember going through those, we're going through the 5 criteria that to make it so to meet Hardy-Weinberg. The last one is natural selection. And so this is just a brief review, but remember, natural selection is the idea that organisms that contain the alleles that are better adapted to their environment will survive more. So it's going back to this idea of being more or less fit. In this example here, we have a dark surface. The dark organisms better blend in, and so they will have a higher chance of surviving to the next generation and being able to reproduce and pass on its alleles. And so we see a frequency change for those alleles over time. And we would eventually see these ones kick the can. And what's the joyous thing? So maybe for the 3rd generation, you would see that it would never go to fixation though. And the reason why is because you have some hidden in your population. And that's the great thing about heterozygosity, being a the heterozygotes, is they are hiding one of those alleles. And this can be good or bad. Again, going back to this idea with incest, in this case, it's not good to be homozygous recessive or to be a carrier, because maybe we don't want that disease. But sometimes it's a good thing. And because, for example, let's say that this environment turns white. Well, now when these two mate, if they do mate, it's all random. They have a 25% chance to make an organism that would be white. It would survive, be able to pass on its alleles to the next generation. Another example of natural selection here is mosquitoes. So this is actually a real life example of what we kind of have observed in mosquitoes. To combat mosquitoes in West Nile, what we did is we used DDT, which is the pesticide. And what they've actually found is that some of the mosquitoes were able to die and some survived. They were some that had an allele that made them resistant to DDT, resistant to DDT. And so they survived. They're now able to reproduce, and we see this happening. And so this is actually a concern, an issue with using pesticides, that make sure that we're using them properly. And this is also another example for antibiotics. So with antibiotics, again, antibiotics attack and kill bacteria in our bodies, but bacteria, they can reproduce and evolve very quickly. When you go to the doctor and you get an antibiotic, they'll say, well, you need to use this antibiotic for 10 days. Make sure you use it completely for those 10 days. And the reason why it's so important that you use it for the complete time that they say to use it is because if you don't, some might survive. Okay. And so if you use the antibiotics the entire duration that you need to, the doctors say you have a higher chance to kill these guys. They can then no longer pass on their alleles and make a population that is all resistant. Okay. This is a concern because some people, they'll take antibiotics when they don't need to, or they will stop prematurely because their symptoms have gone away, even though deep inside they could still have those bacteria still alive. All right. So when we're diving into this, looking at natural selection, there's actually 3 ways that natural selection can affect phenotypes. Okay, and I wanted to just put a plug in this real fast. We have directional, stabilizing, and disruptive. So let's dive in and look at each of these. So with directional selection, what we actually see is, so here in the dotted line is our before, and this is our after. So before evolution, before natural selection set in, we saw a beak size, and this is looking at the finch beaks, a little slightly smaller, but after some selection event, we saw the average change. Again, with these three, it's for polygenic traits. So with traits that are affected by multiple genes, do we see this? It's not just small or big, but we can see a range throughout. Some organisms are right here. Some organisms have beak size right there. Some beak size right here, clear up to very large beak size, and you can see the survivability of those with this curve. So with directional selection, this is when individuals at one end of the curve have higher fitness than individuals in the middle or the end. Okay, and so we can see it's going to shift to one direction. Okay, and I'm not talking about the band, but one direction. Okay, so it could go in this direction, or maybe in a selection event would cause it to go to smaller beaks. Human height's an example of this. So we've actually seen this actually happen. Early, we've actually seen a progressive increase in size in males and in females over time. So this is an example of directional selection. Stabilizing selection is a different one, or another example, where it is what we see is actually one phenotype is obviously more fit, and it's usually in the middle here. So it's when individuals near the center of the curve have higher fitness than at the edges. So what you actually see is these guys would die off, which would then change the population to being more in this range. So it just kind of narrows the curve. An example of this would be birth mass. So birth weight mass. We've actually seen a more stabilizing event there when we look at this. Because if they're too small, think about what that means. So smaller babies have a higher chance of dying. They're not as healthy, I guess you could say. But too big of babies can't get through the birth canal, right? And they're just too big, and you could kill the mother that way too. And so they've actually, through evolution, natural selection, we have seen it find that happy medium, and it's stabilizing at that point. All right. The last one is disruptive selection. This one's kind of fun and very rare. Okay, so this one, make sure you understand. Very rare. Not found in populations very often, where we would see the two extreme phenotypes being more fit than the heterozygote, you could say. Okay. And we call this disruptive selection. So disrupts the one mountain into two mountains. So here is just a quick review of this, where you can see that both dark and white, but gray, or that middle phenotype, would not be a benefit. Stabilizing selection, we see it, the extremes are less fit than the heterozygote or the middle. With directional selection, we either see it go from one phenotype or another in that direction there. All right, last slide. So the last thing I want to talk about is sexual selection. So with sexual selection, this also plays an important factor. It's a type of natural selection, but basically it's the idea here that, and we see this in humans all the time, but we also see it. Some of my favorites are in birds. So with birds, we see this flashy expression. So with the peacock, the male will have this flashy adornment, I guess you could say, to impress the lady. And if the lady is pleased and happy, then that male gets to mate. What we see actually happen, though, is we see a combat between natural selection and sexual selection, because here is this male expressing himself. He is more likely to be seen by predators and get eaten because of that. And so we actually see natural selection taking effect here on what the male will show, and sexual selection of he wants to be as showy as possible, but not showy enough that he gets eaten either. And so we see this battle between sexual selection and natural selection. Yeah, I think that makes sense there, and hopefully that's just a review for you guys. But anyways, that's all I've got for you. And so what you'll do today is, again, there will be a document there with some practice questions just to prep you guys, make sure you guys are ready to do some different problems on the test that are kind of like that. And you're more than welcome to work through those. Well, you're not more than welcome. You need to work through those, but you can use your notes to help answer those problems. Make sure you can do the math with Hardy Weinberg, and make sure you know the different types of like genetic drift and sexual natural selection, that kind of stuff. All right. With that, I'm gonna end this long lecture, and I'll let you guys get going.